All right, so today I'm going to talk about the light reaction. So we have two processes that I'm going to go through. One is the light reaction and the other is the Calvin cycle. So this one we're going to do the light reaction. So what I have drawn up here is remember I refer to them as the spree. The spree is the grana, which is those little discs inside the chloroplast. So we're dealing with the structure inside the chloroplast. So the first thing that I'm going to draw in here is the two photosystems. So we have, again, photosystem 1 and photosystem 2. So and we often do abbreviate them PS1 and PS2. So again, and I always tell my students that it does not mean PlayStation, it means photosystem. Now do also notice that photosystem 2 comes before photosystem 1, and that has to do with their evolutionary development. PS1 came first, PS2 is an add-on. So we're going to start with our PS1, and again, remember inside the photosystems are all those little pigments that we've been studying, especially when we do this in lab. And so this is where we're going to be absorbing the light. So I don't have orange or yellow. So I'm going to represent the light waves with my red squiggles for photons of light. All right, so next, the first thing that we have to do is we're going to take, remember, our water. We have hydrolysis occurring here. So we're going to split water. When we split water, we have a couple things that are happening. And just a little side note, remember that hydrogen is made up of one electron and one proton. And so when we split this, we're not only splitting into our elements, oxygen and hydrogen, but we're also splitting hydrogen. So the electrons are going to go into the photosystem 2. Oxygen is our waste product, and then we're also going to get our protons inside this space. The interior here is called the lumen. So once we get our electron, which is going to replace the electrons lost in ETC as we replace them, that energy is going to give our electron, the energy from the sun is going to give our electron that power boost. And then we're going to play past the electron as we move through our electron transport chain. And I don't think I have any more colors. Oh, I got one here. All right, so as we move and pass the electron, especially when we get to this protein right here, it loses a lot of energy, and it's going to lose that energy because it's going to do work. And that work that we're going to do is we are going to pump the proton. So that is like our proton pump that we talked about in cellular respiration. All right. So what's happening here is we are building our concentration gradient of protons inside the lumen. Once we then pass to PS1, we get another power boost. And then we're going to again go into another photosystem. And this, or electron transport chain, and this is going to be NADP plus reductase. And so what we're going to do here is we are going to take our NADP, remember it's in plant, so we have our extra phosphate, and we're going to reduce it. So we're going to pick up those electrons. So we talked about our final electron acceptor in ETC for cellular respiration being oxygen, and here it is our NADP+, our electron taxicab as I refer to it. So right now what we've done is we have two photosystems, We have this portion right here, which is our ETC, our electron transport chain. We have another one right here, ETC. And so far we have accomplished this. We have picked up our electrons, and so this was all our redox reactions that I keep referring to. And we also have built our proton concentration gradient. Now we have done this for a reason, and the reason why we did this is because we are gonna have our ATP synthase. And remember, with our ATP synthase, we're gonna undergo chemiosmosis, which means we're gonna diffuse our protons down that concentration gradient. And as we do that, we then have enough energy to phosphorylate ADP to ATP. Ooh, helps if I put a P and not a B. All right, so we have two products from the light reaction, and that was our goal. So our goal was to make energy for the Calvin cycle. And the type of energy we made was, first we made NADP+, and the second was ATP. And again, this was all made possible by the sun. So the sun drove the energy for this process. This is why we water our plants right here. We have our waste product oxygen. We split that hydrogen into electrons and protons, pass the electron. We use the energy here to drive that proton pump to establish our concentration gradient. And then we pass the electron again where it gets another power boost. And now this energized electron can be used to reduce our NADP+ to NADPH. Now in the second video, I'm going to show you how we're going to use this energy to fix carbon.